Good afternoon. Welcome, Dr. MGR. Sorry, Dr. Jaculine Pereira. Ma'am is Assistant Professor and Head Department of Aquaculture, Dr. MGR Fisheries and Research Institute. Uh,
Thank you, sir.
this application molecular fisheries. In application molecular techniques in fisheries. Ma'am, you are audible as well as visible, and the screen is visible. You can start.
Okay, sir. Thank you, sir, for giving opportunity. A very good afternoon to everyone. Now we are going to discuss about the topic application of molecular techniques in fisheries. What is molecular techniques? Actually, we are going to discuss about the molecules, the molecular level, that is inside the cell, that is DNA, RNA, and proteins that will give accurate and sensitive, timely diagnosis for any problem. It's a, if it's a pathogen, if it's a genetic problem, or any food products of that's any fishery products can also be diagnosed molecular way. Let us discuss what are the areas of molecular diagnosis. It is applied in species identification through molecular diagnosis, and it is able to diagnose, determine the genetic variations and the population structure about this population. And with this molecular technique, we can able to compare wild as well as natural populations. It will help our assess the assessment of demographic bottlenecks in the natural population, and in some situations the habitat get disturbed, or due to over exploitation the fish stocks get declined. So in that situations, propagation assisted rehabilitation programs can also be possible with the molecular diagnosis. And because of the intensive aquafarming, there is outbreak of lot of diseases. So disease diagnosis with molecular techniques gives accurate as well as rapid diagnosis also possible. Then drug metabolism, nowadays giving treatment or a discovery of the some medicines, that also possible with the molecular diagnosis. And so it is able to give best and appropriate therapy. And coming to the fish processing industries, fish and fishery product industries, qualitative analysis as well as the food fraudulence can also be analyzed both qualitatively and quantitatively. Let us discuss the what are the molecular markers available now. That's allosine markers, that's allelic markers, or then the RAPD markers, that randomly amplified polymorphic DNA, that is PCR based marker. Then RFLP markers. Then single nucleotide polymorphism, a single nucleotide able to give the wide variation in the exons, that in the gene expression, that also can be possible. Then microsatellite markers, again the simple sequence repeats. Then gene expression can also be studied with the expressed sequence stack. Then mitochondrial DNA markers, DNA barcoding techniques. DNA barcoding techniques is one of the mitochondrial marker. Apart from this, cytochrome B can also be used. Then various blotting techniques like the southern blot hybridization, northern blot hybridization, then western can be applied, western blot hybridization that can be applied for the protein analysis or immunotechniques. Then sequencing and micro sequencing, we will these things we will discuss later. Then finally, the advanced techniques, the microarray or microchip. Then this slide shows what is nothing but the DNA. The DNA, as we know, it's a negatively charged one, the negatively charged molecules. So this some of the papers I have given in between. That's that literature, that is molecular markers applied in the different research. Then we will start with the various techniques. First, start with the gel electrophoresis. Electrophoresis is nothing but is under electric current, the molecules getting separated, with giving a under the electric current. The molecules like DNA or RNA or protein molecules getting separated. That is based on the intensity of the electricity we are giving, and based on the matter index we are giving, and based on the charges of the particle. This is the DNA sample loading into this picture shows the DNA sample loading into the gel. This is this picture, the PCR product we can visualize under UV transilluminator. This is in the gel documentation system. We can see this. We can able to take the results of gel electrophoresis. During the gel electrophoresis, actually, what are the factors affecting the mobility of the molecules? That is mainly of molecular factor as well as environment factors. Molecular factors means based on the charge, size, and shape. And the environment factors, nothing but the what is the environment we are applying in the electrophoresis, that is electric field strength and matrix. What kind of matrix we are applying, what's the percentage, that's agarose gel electrophoresis or acrylamide gel electrophoresis, what is its concentration, everything is differ. Then the running buffer, let's say either TAE buffer or TB buffer. This is the vertical section of the electrophoresis, that's a gel. This again, the type of matrix you can use a paper like so then agarose or acrylamide. There is nothing but the matrix, the starch or cellulose phase. Then in the during this picture shows DNA agarose gel electrophoresis results. This is how the polyacrylamide, this reaction actually within the short period we have to cover the techniques. So I am doing somewhat fast. And this how the polyacrylamide gel, this chemical structure shows the polymerization of polyacrylamide gel. Then coming to the blotting techniques, actually of mainly the different blotting techniques are there, mainly southern blot, northern blot, and western blot. Southern, actually, the name of the scientist, he did DNA blotting to honor his name. It was given for that. That's a southern blotting. Then afterwards, for the RNA or northern blotting, then the protein western blotting, the name was given. Here in the southern blotting, there are two important features of hybridization. First, the reactions are specific because the probe, what is the probe? Probe is nothing but a single stranded nucleic acids that will seek complementary sequence where is exactly there. So it will find out the one molecule in the target, then there it will bind that the complementary to that of the complementary sequences. So in the southern blotting, DNA fragments actually fragments of the large strand of DNA will while DNA extraction will be getting the large DNA. So that from the DNA we have to make it into small frequent fragments. That is with the help of the restriction enzymes. The restriction enzymes are nothing but they are the biological scissors that will cut at the specific site of the particular gene of our interest. So it will make this restriction enzymes will cut the DNA into specified size based on the specific site. It will make various fragments that can be separated with the help of Agarose Gel electrophoresis. Then it can be transferred into the membrane, nylon membrane or nitrocellulose membrane. Then the probe, I already said what is probe. It's nucleic acid, single-stranded nucleic acids that will seek where exactly complementary sequence is there. There it will attach. Then we can detect in the film or fluorometer or with auto radiography. Then coming to the northern blotting. Northern blotting is nothing but RNA blotting. And this is just like that of southern blotting. Here the difference is RNA is the starting material. Like that western blotting. It's a protein. Well, instead of protein, that is instead of DNA or RNA, here it is protein. But here the gel matrix, we have to use the polyacrylamide gel, that's of SDS, sodium dodecyl sulfate, that denatured acrylamide gel we are using, and that will for the give that for the purpose of immuno diagnosis. Then after that, after kind of any blotting techniques, will go for the hybridization with the probes. That if it is DNA, it is the starting material. That's DNA hybridization, or if it is RNA or DNA RNA hybridization, or RNA, that is depends on the our purpose. And again, it is depends on the temperature, salt concentration, and the buffers what we are using. And again, labeling also differ. Label without the labeling, we don't know, we cannot detect the hybridization. So for that purpose, we will go for radioactive labeling or fluorescent dyes or enzymatic labeling, so we can detect the hybridization. Then coming to the another techniques, that is PCR, polymerase chain reaction, that we all know, aware of all this technique. That is, it was discovered by the scientist Carey Mullis during the year 1983. He is a chemist, and he got Nobel Prize for his work during the year 1993. This slide shows how the PCR reaction is taking place in a short within this one PPT is able to, you can able to see the first single strand DNA. So how this denature, then annealing, and the extension, that we will discuss in the next slide separately, give some for emphasis. This is the three steps. While discussing about polymerase chain reaction, there are three main steps. That is denaturation, annealing, and extension. Denaturation, actually, what is happening, and that is here the DNA strands getting separated at high temperature, at a temperature of 94 to 98 degrees centigrade, the DNA strands getting separated into two single strands. Here we can see that then next step is the primer annealing or priming. Priming is the primer will seek where exactly complementary sequence present in the target DNA. There it will bound together. So prime the polymerase reaction. Then the next step is polymerization. Since this polymerization reaction was carried out with the help of the enzyme, DNA polymerases. The polymerases, actually, there are different kinds of polymerase enzymes are there. Though the enzymes, different enzymes are there. Tag DNA polymerase is one of the best enzyme nowadays in practice. Because why it is tagged DNA polymerase is the best one? Because all the enzymes are proteins. Proteins get denatured at very high at high temperatures. So once it is denatured, it cannot become the normal one. It loses its activity. So because of that problem, avoid all other polymerase enzymes. Only the Taq DNA polymerase is preferred one, because it has extracted from the bacterium Thermus aquaticus. It is from the hot spring. So this enzyme can able to withstand even at very high temperature. There is 94 to 98 degrees centigrade. Can also be that Taq and polymerase enzyme can be able to withstand it at high temperature. And this enzyme only by undergoing the process of that will recognize the primer, and that will take the that makes the complementary copy of the template DNA and into multiple copies. So this slide shows there's a program of how we can give PCR programs. The step 1, it has given initial denaturation. The step 2, that actually this program depends upon a gene of our interest, which gene we want to polymerase, we want to amplify which gene, and based on the gene we will decide the annealing temperature, that's melting point, and it depends on the GC content of the particular gene, and so it will denature. So then at high temperature always it will get denatured. The steps and all we have already discussed. So the step 2 in the program, the required number of cycle has given. So that is it can be 30 cycle, it can be 35 cycle. That is depends what is the program we are giving. It will work on. Then after one cycle over, and again coming to the step 2 till completion of that is 30 cycle or 45 cycles. What is how many is the number of cycles we have given? Then the third step is the final extension. That is final extension that is remains for 72 degrees centigrade. It can be, it need not be 7 minutes alone. It can be 10 minutes or 7 minutes. In this program, it has given only 7 minutes. Then after that, once the PCR reaction is over, means we can keep it under refrigeration, store the samples at 4 degrees Celsius. That only given in the program. So this is this picture shows from a single strand. See, from a single double strand, from one double strand, we can able to multiply into millions of copies. That is 1 into 2, 2 copy. Then 2 into 4. The cycle 2, you can see here 4 copies, and cycle 3, 8. Like that, 2 power n number of copies will be produced. Then what are the optimizing PCR components? We have to see that. First, we have to see the starting material. That's a raw material of that's a nucleic acid, whether it is DNA or RNA. If it is a RNA, we have to go for one more step, that is reverse transcription steps. Then we have to see the polymerase enzyme, and primer and buffer. All these things are the to optimize while going for raw material can be of any, that's our target DNA. Raw material is nothing but target DNA. It can be of any organism. It can be from any organ or tissue or cell. What is the best starting material? And while collecting blood, and also some products, some kind of inhibitors also there. So that in case of a blood, the heme is the inhibition product. So that product, the heme can be removed while going for the blood sampler. And then like that, good quality genomic DNA, we have to see that the quality of the material, the raw material also, we have to make sure it's of a good quality. Then coming to how PCR is powerful, we know that, but because of the corona period, many of them, even our people, layman also know about what is PCR, what is RT-PCR. So because that is diagnosis was carried out with the RT-PCR. So PCR is such a powerful technique. That's why this technique is employed. So here the template DNA need not be highly purified. Says if you are taking any bacterial colony, just boiled colony that able to give the DNA. So that is not highly purified is raw material or target DNA is not necessary. Then the PCR product can be digested with sometimes that it can be used from the digested product that can be digested with the restriction enzymes. Sometimes we can use the raw material or a target molecule from the sequenced product or from a cloned product. So, or even from the sperm or any kind of cell. Then where it is applied? It is applied generally in not in our fisheries field. In general application, going for the general application, it is applied in the paternity testing, and it is applicable to the mutagenesis, or it is able to quantify because RT-PCR level, that is quantify the gene of expression. And so from the unknown, we can unknown gene also we can go for with the analysis with the known one. And forensic science in crime and all applied, but in our field it is applied in the food fraudulence. It is applied so one species is replaced with another species, that can also be possible. Then industrial quality control and say sequencing also. So the PCR followed by sequencing method is applied. So this method is applicable. These are this is a paper with different disease diagnosis that PCR applied. And then next technique is RAPD. RAPD is nothing but randomly amplified polymorphic DNA. So this technique involves this is also a PCR method. Here, what is the difference? Random primers are applied, not the specific primer. Actually, PCR we have to use the specific primers. The primer is specific for any specific genes. Then coming to the RFLP, RFLP is actually what is the difference here? We are using the one step that is first we'll have to go for DNA extraction. Then DNA once extraction will then have to go for treatment with the restriction enzymes. The restriction enzymes I already discussed. Restriction enzymes are nothing but biological scissors. It will cut at the specific site. So because of the digestion with the restriction enzymes, we can able to produce different fragments. So that the different fragments can be separated with the Agarose Gel electrophoresis. Once it is Agarose electrophoresis over, we can go for the blotting technique, so that the DNA present or nucleic acid present in the gel can be transferred into the nylon membrane or nitrocellulose membrane. Then it can be transferred, exposed to the photographic film. Then we have to go for labeling with radioactive isotopes. Then we can go for autoradiography. This paper also used various, that is RAPD PCR, then RFLP, specific. Then PCR again, already said that the PCR based technique or molecular techniques applied in the fishery products also for the authentication of species. So this is a PCR gel RFLP pattern. Coming to the AFLP procedure, AFLP is nothing but for unknown species or unknown gene, also we can go for with the AFLP. What is the AFLP? Actually, this amplified fragment length polymorphism. So here the genomic DNA digested with a known restriction enzyme. Previously, in the polymerase PCR and all, without we have discussed PCR as well as RFLP also. RFLP, there is no amplification. But here, after digestion with the restriction enzyme, we have to go for amplification also. But immediately after completion of restriction digestion, the fragments should be ligated or linked with the adapter molecules. The adapter molecules that will ligated or sealed or added bound with the fragments. Then next step, the normal PCR, that is PCR primers and other process under doing. So after that, we will have to go for the electrophoresis under. So this paper also shows how in the AFLP is used. Well, this is one of the gel. This gel shows AFLP bands. This is a single nucleotide. Next technique is a single nucleotide polymorphism. So in this picture, you can see that here the CG in this picture, but here it is modified. One sequence is changed. If there is a change in a single nucleotide, instead of C, it is T. Means so C always complementary to that of G. Cytosine always is complementary to that of guanine. So adenine. So once C is replaced, means again A replaced with T, means complementary sequence have it. So that what is given here, that is that indicates point mutation. So it's a single sequence change in exons that leads to various drastic change in the gene expression. So this is also another paper with polymorphism markers are used. Then the next technique is DNA sequencing. The DNA sequencing method actually mainly of two methods, methods of DNA sequencing. There is the first method was done by the scientists Maxam and Gilbert. Then followed by the later phases, Sanger's Dideoxy method, or that is now it's currently also we are using Frederick Sanger. He has actually he first sequenced the amino acids, the protein sequencing during the year 1958. But in the later phase, he did dideoxy DNA sequencing during the year 1980. So this is dideoxy molecule. So this it will replace the our deoxy molecule, deoxyribonucleic acid, DNA. Dideoxy replaces the DNA molecules. So here. This in this step principle, the partial copies of DNA fragments that is made with the DNA polymerase. So that is first here that terminate that the dideoxy undergo the process of termination of the amplification. So that we will be getting the different single ACTGC that 4 nucleotides base replaced with a dideoxy NTP. NTP is nothing but N instead of N, ACTGC. ACGT. So that is dideoxy product will be given. That is chain termination process has done, and this can be separated by gel electrophoresis. Then we can read. So here this dideoxy, how it is getting. So because of the replacement, if it's adenine in the sequence that is replaced with this A is replaced with DDA ATP, like that. T means DDTP, like that. So here you will be getting that the various sequence with the different color patterns. So this, if this picture shows if this is the single strand of DNA to be sequenced. This is our PCR product or any product. If you are sending for sequencing, if you are giving it for sequencing, so this can be. This is the first reading from 3' end. Here, this is the EA. That's adenine in the sample means here the complementary sequence thymine is produced. Again adenine, thymine. If it is cytosine, guanine, like that it is there. So here you can read the different sequence with the different color patterns of the band. So that also with the during electrophoresis getting the pattern of bands. We are comparing the different sequencing methods, Sanger method and the previous method, the Maxon and Gilbert method. Maxon and Gilbert method is a chemical method. Here, this is a enzymatic method. The second Sanger's method only nowadays in practice, it requires DNA synthesis and the chain termination also takes place. Here, the other method, it is not like that. The pyrimidines get degraded, and with the help of the chemical compound, the piperidine is the chemical compound used for the degradation of nucleic acids. This is paper applied DNA sequencing. Many then coming to the mitochondrial DNA markers. Mitochondrial DNA markers are actually mitochondrial markers are actually, that is we can use any kind of cell, mitochondrial cell, because I all know that mitochondria is a powerhouse of the cell. So it is a maternal inheritance. Only from the mother it will be inherited to its offspring, and this DNA have 37 genes, and usually we are going for the DNA barcoding, means with cytochrome oxidase subunit 1 gene. And in our laboratory we did DNA barcoding of loaches, then we barcoding of flying fishes and cardinal fishes, and we have deposited 30 DNA sequences to the GenBank, and we got accession numbers for that. And this paper also shows the various molecular markers in the applied in fisheries field. Now let us how to choose. Now we have discussed about the various markers and its applications. Actually, choice of markers, how to choose the various markers. That is depends on the. We have to consider the what are the points while choosing the markers. That is depends on the rate and mode of evolution, and the mode of inheritance, then mode of expression, availability of the sample, sensitivity, availability of the markers, then multi-locus or single locus or, and cost. We have to work out the cost also. And coming to the various technique, well, also based on our purpose, we have to choose the various RAPD, the various molecular markers. If our purpose is species identification, we can go for RAPD, AFLP, SSR, and allosines. If our case is a strain identification, so any microbial samples, particular bacteria, we want to see the different strains, means we can go for AFLP, SSR, or RAPD, RFLP, then allosines. If we want to go for monitoring genetic changes in the stock, if there is stocks undergoing the genetic changes, how to monitor in such conditions, we can go for mitochondrial DNA and then allosine, SSR, like that. And like that, various purpose, the parental assignments and reproductive conversion, we can go symbol, that SSR symbol sequence repeats, like that genetic diversity, AFLP, SSR, RAPD, mitochondrial DNA, that's an allosines. Again, brood stock management, again we can go for SSR. Then identification of genetic variability within the stock also, we can go for mitochondrial DNA, SSR, allosines. Then we want to go for pedigree analysis. We want to see the history of the pedigree analysis in the selective breeding aspects, that condition we can go for mitochondrial DNA SSR. Then genetic manipulation, if there is any genetic manipulation happen, that also possible with SSR and allosite. Then we want to go for a genetic mapping, genome mapping, or a quantitative trait local detections. We can go for SSR, RFLP, AFLP, and SLP. So, coming to the PCR products authentication, there also we can go for RFLP, SSR, the various techniques, PCR-based techniques, COI, cytochrome oxidase, COI. Once we have amplified, say, cytochrome oxidase up into one gene, again we can go for PCR, RFLP. PCR product can also be used for PCR, RFLP method. Then we all know the next techniques is quantitative assay, that real-time PCR. Real-time PCR, if the RNA is the starting materials or a target molecule, or our target gene of interest is RNA molecule, so first we have to go for isolation, extraction of the RNA. Then we have to go for the RNA. It's that instead of thymine, uracil is there in the case of RNA. So that RNA you have to undergo the process of cDNA synthesis. cDNA synthesis nothing but RNA will be converted into complementary DNA. So that step, the additional step to that of the PCR. So next PCR amplification only. Here in the real-time PCR, we can able to possible quantitatively. So this graphical representation will give in the real-time PCR the quantitative analysis. This is this flow chart shows how the real-time PCR is carried out. First, RNA sample, that total RNA will be getting. Then we will add the DNA treatment. That is because there should not be any DNA contamination. So that then we will be for that we are adding DNA enzyme to digest any contaminated DNA is present. So after that, the RNA alone, isolated RNA will be getting. That is taken for the steps reverse transcription step. Once RNA is converted into cDNA, that DNA will be taken for the PCR analysis. That is quantitatively we can able to diagnose. So this method is applied for the any gene expression studies, metabolomics, any physiological activity, or any gene expression studies, a particular gene or protein, the particular gene expression, particular amino acid is produced or particular protein is produced. That can be so before that the particular before going for the protein synthesis, mRNA is the transcript. So that can be possible with the real-time PCR. So for here in the flowchart tissue sample, first take isolation of tissue sample, then from that we are extracting DNA, then cDNA RNA will be converted into, we have to convert it into cDNA with the reverse transcription system, then PCR. Then this PCR product can able to give quantification, and that can be test for that is statistical mode. Next, advantages. What are the advantages? Now we have discussed about various techniques and various molecular techniques. Then while going for molecular techniques, what are the advantages? Why we are choosing this? Because it is highly sensitive and highly specificity, and we can. It is speed is quicker than any traditional culturing method, because traditional methods, if you want to diagnose any microbes, means we'll go for microbial testing. First total plate found, then selective plating, confirmatory test. That is, we will follow the Burgess manual steps in the Burgess manuals. And so first, it will take initially about a fortnight, that once 15 days period. So nowadays, it is with this molecular techniques. Otherwise, we'll go for immunotechnique. Immunotechnique also arising anti-serum and all. It will take time. So to speed up the detection system, it is possible with that is molecular testing, and it is simplicity also. It is easy, simply if you can. Now it's again automation is there. But though it has a lot of disadvantages, there is some disadvantages also, because it is expensive one, and it must have good clinical data about the particular organism. Sometimes we will miss new organism unless we are going for sequencing. If we are the PCR product, if we are not taking further step, sequencing is not taking, means sometimes we will go for, we will miss some new organism. And sometimes there may be amplification may have some problem, and that is it will amplify some troubleshoots in the that mixed cultures will give some problems, or some contamination will be there. So that also leads to some problems, and because PCR is too sensitive, so it has less clinical value also. Then coming to the conclusion, molecular biology or molecular techniques applying in the, starting from the aquaculture, then to the products. It's a vast field, and nowadays in our medical research also applied, but in our field also it is for the disease diagnosis. It has promising challenges. It is able to give promises in challenges in the disease diagnosis of the pathogen. So importance of the research on the molecular markers, it is had to improve because of the molecular markers of various research on the molecular markers. It has enhanced the computational power, and it is able to derive to obtain a large data for the researchers. And since the innovations provide, and this innovations provide concerning many population bottlenecks and migration of the species, and many gene is possible, and it is applied in the field of disease diagnosis, therapeutic measures, prophylactic measures, and again it is applicable in the quality aspects also. Quality, that is quality of the particular food products, and pathogen detection and all, and food fraudulence also possible with this. I'm completing my presentation. Any doubts, please? Because with short period we have completed many techniques. Now it is open for the discussion, please. Now it is open for the discussion. No question? All are here?
Participants, if you have any questions, queries, or you want to interact or thank ma'am, please go on. Any questions? I think, ma'am, there are no questions.
Okay, ma'am. Thank
Uh,
you.
Thank you, ma'am. Thank you on behalf of NADCL. Thank you on behalf of candidates. It was really a very interesting and very interactive session. Hope all the participants have benefited with it. Thank you so much, ma'am.
Thank you, ma'am. Thank you for giving opportunity. Thank you for NADCL and CFT organizing this program. Thank you, ma'am.
Thanks again.
Hamida Khanam, sir.
We will leave the session now and join on Monday again.
Oh, okay, ma'am. Shall I join?
Yes, ma'am. Yes, ma'am.
Okay, thank you. Thank you, ma'am.
Thank you again.